let me make up a pedigree, and then we'll see if we can figure out how to turn it into a chi-square related question. Let's see, 6 offspring. Wait a second, what the heck are we doing? Okay. 6 offspring and a cross. And I'm going to pick an inheritance pattern. Okay, let's stop there. This should be a good example, I think. What's the inheritance pattern?
Okay, well, you have recessive, dominant. What's that one with the C? That's the word.
Cytoplasm.
Thank you.
Okay, so which can we rule out?
Cytoplasmic.
Cytoplasmic, because mom would pass the disease or phenotype to all of her children if it was cytoplasmic.
Yeah, and it's only 2 that are demonstrated. And you could rule out Y.
Why?
Because the dad is not expressing the gene, although he could be a karyotype, but sorry.
Well, there are no carriers for Y-linked.
Yeah.
So if you've got the mutation, you get the phenotype.
And he's only passing it to
More importantly, who's got the phenotype?
mother.
Yeah, who doesn't have a Y chromosome.
Yeah.
So it can't be Y-linked. So yes, it would be passed from an affected father to all of his sons and never show up in a female. Okay. Now, could it be dominant?
It's possible.
Okay, so possible that it's dominant. So that leaves us with recessive.
Okay. Well, since the mother already has the character,
Mm-hmm.
and she's passed it to daughter, which would get half. These are X, right? From her.
Yeah, so if it's
possible that the father could be a carrier because she's expressing all of the gene and not just part of the carrier.
okay, so if this is recessive, what you're saying is mom is minus minus.
Mm-hmm.
And and we know that for a recessive trait, if it shows up in 2 generations, that means that the other parent has to be a carrier, so that these kids have the opportunity to get 2 copies of the mutant allele. Mm-hmm. So then the question is, for an X, can males be carriers for X recessive traits? So male has 1 X chromosome.
Mm-hmm.
So for him to be a carrier, that means that his 1 X chromosome would have the mutation.
Yeah. Unless it's a freak mutation. But that's less likely.
Yeah. So the deal with X-linked recessive mutations is there's no such thing as a male carrier. If the male has the mutant X chromosome, they show the trait.
Mm-hmm. So father doesn't show the trait, so it's impossible.
So it's not X recessive, because that father would have to have to show the trait. Okay. What would be true if this was autosomal recessive?
That would be carried from not just one individual, but many.
What's the genotype? What do we know about the male? So it's not X recessive, but it could be autosomal recessive.
That if he carried it, then he'd have to pass the gene on to his daughters.
Right. And sons.
And
Autosomal. So nothing about sex anymore. If it's autosomal recessive, then dad's a carrier.
Yeah.
Okay. So it could be autosomal dominant. Let's see, X dominant. Could be X dominant. Okay. Could be autosomal recessive. So how do we determine which of those three is most likely? If this is dominant,
Mm-hmm.
let's see, this is generation 1, generation 2 for nomenclature. So the generation 1 female, what is her genotype?
I'd say it'd be negative and pressures. Kind of like
That is she homozygous for the mutation or heterozygous?
hetero.
Okay, why heterozygous?
Because if she just split half of the gene, she'd most likely be able to carry it to multiple children.
Well, she did have...
She didn't have 2 examples of it.
Right, so she got 2 out of 6 kids have the disease. So that tells you... I'm still trying to get you to tell me why that means that she's a heterozygote. What would be true if she was a homozygote for the mutation?
She'd have the same gene, so it would be same, so she'd end up only distributing... Oh wait, I messed up on that. Ha ha ha. Okay. So if she was at homozygous,
Yeah.
then she'd have half. So if the dad had the other half and was a carrier, then it's possible the children could get half of the mother, half of the father, and end up having the gene.
Oh, but we're talking about dominant, sorry.
Sorry.
So if it's autosomal dominant or X-linked dominant,
Mm-hmm.
what's the mom's genotype?
It would be I think I'm confusing
So
myself.
look at her kids. So if this is dominant, we don't know if the mom here is heterozygous for the mutation or homozygous. So she could either be plus over minus, or she could be minus over minus, with minus controlling this trait, right?
Yeah.
How do we determine which of those two is her genotype? I can tell you one of those two from the data we have right now. We can figure out which of those genotypes she has if this is a dominant inheritance pattern.
Negative?
Which one?
Negative? No.
Well, look at her kids. If this is dominant, one of those two genotypes of the mom is incompatible with this pedigree. What fraction of her kids got the disease? Well, third, but which is less than all of them. So what's mom's genotype? Because if her genotype was homozygous minus, what would be true?
They don't have to show it.
All of her kids would have the phenotype if she was minus over minus. There you go.
I wasn't seeing it. I'm sorry.
That's okay. So for this mom to have kids that are unaffected, if this is dominant, she has to be heterozygous. Because if she was homozygous, then all of her kids would be filled in, because they'd have to get a minus allele from the mom. Okay. So we're going to assume that if this is dominant of either type, that the mom has to be heterozygous. Okay, if it's autosomal recessive, what's mom's genotype?
Yeah.
Do this in blue.
It'd be homozygous.
So, generation 1 mom has to be, if it's recessive, if she chose it, she has to be homozygous recessive. And then what has to be true about... Yeah, go ahead. For some reason it stopped recording there at some point. Anyway. Yep. Transcription. We've got DNA molecule. We're going to wind up producing an RNA molecule. So it's going to be single-stranded, and it's going to have U's instead of T's. So how does transcription initiate? What's the start? What's the enzyme that actually does the reading of the DNA and the making of the RNA molecule?
RNA polymerase.
Yeah. So somehow we have to get RNA polymerase to the transcription start site. So there's the transcription start site. Here's RNA polymerase. So it's going to bind to the DNA and start transcription there. And that's the role of proteins that bind the promoter, which is sequencing the DNA that's upstream or 5' of the transcription start site. And depending on which type of organism you're talking about, prokaryote or eukaryote, that's any one of a number of different proteins that bind a specific DNA sequence in the promoter, and then those proteins positively, physically interact with RNA polymerase to hold it at the transcription start site until it actually starts transcribing. So then what RNA polymerase does is it denatures, it unwinds the DNA so that it can actually read one of the strands. So there's RNA polymerase at the transcription start site, and it's going to move this direction with its bubble along the DNA, and it's going to eject out of a pore in the protein the messenger RNA starting at the messenger RNA's 5' end. So like all DNA and RNA synthesis, it starts at the 5' end, and polymerase adds, any polymerase, DNA polymerase or RNA polymerase, adds nucleotides onto the 3' end. Right, so the 3' end is there, and the way that RNA polymerase knows what nucleotide to add is because it's reading the DNA sequence and it's sticking in the complementary nucleotide. So what's the next nucleotide RNA polymerase is going to add to the 3' end? Here. So what's complementary to G?
C.
C. So it's going to add a C. And the next thing it's going to add is
A U.
U. And then as polymerase moves from left to right, then it'll add an A and a G, and it'll keep adding on to the 3' end. Okay. So that's transcription. We've done initiation. That was the promoter binding, proteins binding the promoter, recruiting RNA polymerase to the transcription start site. Now this is the elongation step, where RNA polymerase is reading the template strand and producing an RNA molecule. So, which of these two strands of DNA is the template strand and which strand is the coding strand?
The top one would be the...
Go with your gut.
I know the bottom one is the coding strand.
No, not always.
Not always.
You're right in this case, but the bottom strand depends on how you draw it. So why do you know that the top strand is the template strand? It's because the template strand is the one that's actually being used as the template for transcription. It's the one that RNA polymerase is doing the base pairing with to make sure that it's adding in the right nucleotides. Okay. So what's the sequence that I haven't drawn yet? The coding strand sequence for these 4 nucleotides. What's it going to be? Starting at the left.
This one is C.
Yep.
U. No, A. No.
You get one more choice.
T.
Yeah. Because the G and the C will base pair together, the A and the T will base pair together, the T will base pair with
The A.
A,
And the G.
G. Right. So now we've got DNA sequence that when this is double-stranded, when RNA polymerase moves past, these will base pair with each other again. Okay. Now, 2 more questions. One more question and 1 point. Note that the sequence from the 5' end of the RNA molecule and the coding strand is identical. Right? CTAG, CUAG. Okay? That's why there's this rule that whatever the sequence of the RNA molecule is, that's the same sequence as the coding strand. That's how you know which strand is the template strand or the coding strand.
Okay.
The other point that's critical here is, what's the polarity of the DNA?
We'll go from 5 to 3.
So,
No,
top strand,
3 to 5.
top strand turns out to be 3' here and 5' here, and that means the bottom strand is 3', 5'.
Oh, yeah.
Now, how did we know that? The upshot, by the way, while you're thinking about that, although I'm going to be talking, so you're not going to be thinking about that, the upshot is that RNA polymerase is reading the template strand. Note, template strand is the top strand. RNA polymerase is reading the template strand from its 3' end to its 5' end. That's not super important to remember. You don't have to memorize it. But the point is that if you synthesize molecules from their 5' end to their 3' end, and this is the template strand, that means, what do we know about nucleic acids when they're base paired? 2 strands. They're parallel or they're anti-parallel?
They are anti-parallel.
Right, like here's anti-parallel. The DNA, where it's not unwound, is anti-parallel. The same is true of this DNA RNA molecule. They're base paired right here. And so if RNA polymerase is generating RNA from its 5' end to its 3' end, there's the 3' end of the RNA. So it's right next to 5' end, which means that the 3' end of that is there. So you've got base pairing here between the DNA and the RNA while RNA polymerase is making the transcript. So these have to be anti-parallel with each other. That's the initiation of transcription, elongation of transcription, and then termination.
Okay.
Where does this process stop? So RNA polymerase keeps moving down these molecules until it reaches, down here there's going to be a backwards, AAU, AAA. I was thinking about the other strand. That's the poly A signal sequence. So when RNA polymerase reaches that specific set of 6 nucleotides, then a whole set of processes occurs that causes it to stop reading the DNA.
So on the test, are you gonna ask like transcribe this is translate this strand to the mRNA, or like how can you ask that question about this concept?
That's a good thing to think about. You should have thought about that last week and asked and written me a question to put in the mock exam that's like that, because I'm not going to tell you always what sorts of questions I'm going to ask on the exam, but I could say here's a DNA molecule of a particular sequence. I could say pretend you're pretend you're RNA polymerase and you're going to transcribe this. I could.
Can you show an example? Another example. Just so I can actually see it.
Depends. Do you have other questions? Because that would take me a while to do. I'm happy to, but
I mostly want to see that one, so I'd rather see that than ask my other question.
and we're still trying to follow up on the thing we were just working on.
Yeah. because that was pretty much what I wanted you to go over on the Zoom appointment. Oh.
Okay. So DNA. We've got
It's okay.
okay, so there's a really horrible sequence of part of a chromosome. TA, and then there's 20 nucleotides, and then there's a specific sequence here, and now you play RNA polymerase. And after that, play ribosome. So it's a eukaryote. So transcribe and translate this chromosome. I've given you a bit of a hint here that this is the transcription start site. Because you don't ever really know, it's about 20 nucleotides downstream of TA. That's where TATA binding protein binds. So you'd expect in the promoter, which is everything upstream of the transcription start site, there's going to be about 20 to 30 nucleotides upstream of TA that TBP binds. That's what brings in RNA polymerase. Right? So it physically binds to TATA binding protein, RNA polymerase, and then it starts producing a transcript. And let's say the bottom strand's the coding strand. So I've given you the coding strand DNA sequence. So what's the sequence of the transcript?
So you always do the AUG? No. What is it?
No, this is the sequence of the coding strand of the DNA.
Okay.
So the mRNA has what sequence?
Is that a parentheses or a C?
That's a C.
Okay, just checking.
There's no parentheses in DNA sequence. I'm kidding. I mean, I'm not kidding. There really isn't. But so is the RNA sequence going to be the same or complementary to the coding strand sequence?
Complementary.
It's going to be complementary to the template strand, because the template strand is what RNA polymerase is matching sequences with. So coding strand and RNA sequence are identical, except for what exception?
Oh my god.
So the messenger RNA molecule that produced by RNA polymerase as it moves down this gene is CGAG A what?
U.
There's the exception. AUG G. Oh, that should have been a T.
I could change the
UAG
U.
AUAA. Oh, wait, that's not a nothing. And then that should have been a T. ATA. So AAUAAAA CGUG.
So why is it converted to an A instead of a T? If it's,
No, that's.
Because you had a U and then you changed it to a T.
Oh, because that's
So
DNA.
Okay, just checking.
Yeah, those should have been. These should have been T's because I was running a DNA sequence.
Okay.
go blame on the professor. Totally fine, because I was thinking RNA when I was writing DNA, and things happen. See, we're all human. Okay, so there's an mRNA molecule. It's had a 7-methylguanine cap attached. And then, is there a poly-A signal sequence?
Yes.
Well, there should have been. It's close. That should actually be, if I had done that correctly, it should be AAU, AAA. So that means about 50 or 70 nucleotides or something downstream of that, poly-A polymerase would add the poly-A tail. But that's unimportant because all of that is in a 3' untranslated region. So that was play RNA polymerase.
Mm-hmm.
And by the way, we haven't done any splicing. That's fine. I did not happen to design any introns. I might have unintentionally, but let's assume that this is a gene that's not spliced. I would tell you that on the exam if you had to know that. So now, ribosome. You're going to translate this. So that's transcription.
Mm-hmm.
This messenger RNA gets sent out into the cytoplasm. So what's a ribosome going to do?
Convert.
This is what we talked about in class today.
I know.
So small subunit's going to bind the 7-methylguanine cap, and it's going to scan down the transcript and find which encodes methionine, and then it's going to read. So it's reading from the 5' end of the messenger RNA. It binds the cap, which is by definition at the 5' end. So it's reading 5' to 3', AUG, and then what happens? Ribosome keeps moving. Or it's GGG.
That's when you gotta look to the coding.
Yeah, better look on the codon table. That happens to be glycine. And then UAG, which is a stop codon. And then the ribosome falls off of the messenger RNA molecule. So that's an example of a DNA sequence that encodes a transcript that gets translated to produce a 2 amino acid long protein.
Would you mind if I take a picture?
Of course not. So the important points are the DNA molecule encodes all of the sequences that are necessary to control transcription.
Mm-hmm.
Both its initiation here in the promoter and also its termination. So the poly-A signal sequence has to be in the DNA itself. And then it's the start codon and the stop codon that are, if you look up here, those are in the DNA sequence, of course, because they're part of the messenger RNA sequence. Those are the things inside the gene that control translation. Mm-hmm.
So we don't do anything with these ones at all? Nothing?
Nope.
Okay.
That's called the 5' untranslated region. Remember, we've got a 3' UTR down there. That's the part that's to the right of the stop codon. Mm-hmm. Everything that's to the left of the start codon is also untranslated, right? Because translation only occurs between the start codon and the stop codon. So there's going to be part that's untranslated because the ribosome reads past it before it gets to the start codon. Mm-hmm. And then there's going to be a part that doesn't get translated because it's beyond the stop codon.
Okay.
So in terms of an equation where you have like this and splicing is added to the equation, how exactly would the structure of it be the same, or because it has splicing in it, would you have to alter the positions of where you have the stop codon versus where you
Splicing will occur between, well, let's see, I don't know if I can see that. It's complicated and it differs,
Okay.
which is horrible. But splicing is incredibly complicated.
I know. That's why I asked.
Yeah.
So, how would you want us to explain it then if splicing has occurred?
Well, what do you look for? The thing that you really need to know is what sequences do you look for to know that splicing might occur?
Changes in the overall sequence? Well, no.
And there are particular nucleotides that tell you where introns are, the parts that get spliced out. Those are also in the DNA. So what is that sequence? Every intron starts with a G and a U. And ends in an A and a G. And every intron has an A somewhere in between the G, U, and the A and the G. But you never know if a G and a U and an A and a G are going to get used as splice sites. That is, if they're going to be spliced out or not. We discussed that in class. That transcription or splicing is complicated. Sometimes these get spliced out, sometimes not. So I tend to shy away from them on. I do have questions about introns, but not sort of actively. Here's a gene, show me all of the different possible splicing patterns, because it's complicated and takes up a lot of time. So you can expect questions about splicing, but there would be directions given to try to minimize the amount of
Work involved.
alternate. Yeah, exactly. Because it's 50 minutes and I've got to ask questions about a lot of different topics.
So,
If there was a question about splicing.
So how would you recommend practicing that then?
Knowing what we talked about in class, going back to that class where we talked about introns and making sure you're comfortable with basically knowing the sequence, which you can always look up during the test if you want to, but the sequence that identifies introns, and then what happens after you splice. That is, what are the resulting molecules after, if this gets spliced out, what does the resulting molecule look like? So for example, if there was an exon here, exon 1, and exon 2 is over here, and those were some nucleotides in the exons, 4 on either exon. Then after splicing, it removes the G and the U to the A and the G. So what you wind up after splicing is a transcript that still contains HEAG, and then it's connected to the next exon. So you've got exon 1 and exon 2 physically stuck together with the intron having been taken out.
Okay. Yeah, I remember that in class. Okay. So that one was splicing.
Yeah.
Okay.
Yep. Yeah. And then you get into the complexity of, is the intron spliced out or is it not? And that's why I just want you to know the basics of intron is encoded by the DNA. It might get removed, and what the resulting product would be if you took it out. You stick exon 1 and exon 2 together.
Yeah, we did exercises like this in class. Okay. So just need to go back to that.
We do not have any genetic distance question. Like genetic distance and...
I don't know, was that
Yeah.
material we covered since the last exam?
Yeah, I think so.
Yes. So yeah, we spent...
2 weeks, 3 days?
Yeah, we spent quite a bit of time on that. So we've talked about linkage, recombination, chi-square test, mapping, pedigrees, and the central dogma. So it's basically linkage and mapping, chi-square test, pedigree analysis, and then transcription and translation.
So it's anything from, technically from the 6th to the...
Right. Till now.
to now. question for this too.
Hang on just a second. Do you also have questions?
I just need to talk about my
Form? Can you step out in the hall for a couple of minutes
Yeah.
while I do that? Just so I try to address everything. And then, but yeah, if I've got more time after that, I'm happy to answer more questions. Thank you.